So our next speaker, we've heard him introduced already by Dr. Fickey, is Dr. Marcus Bamman from University of Alabama, Birmingham. He is a national leader in this field. As I was myself learning about this field, I was about a year and a half ago, there was a Time magazine special edition about exercise, and Dr. Bamman was quoted in that, and he's been nationally known as an expert in this field. He directs the University of Alabama Center for Exercise Medicine. He's been a prolific researcher. He's been spent some time with the NASA at working with the space shuttle astronauts and the effects of exercise as it relates to what they doing. He's large number of scientific articles, currently doing research that's funded by the Department of Defense and the Veterans Affairs Agency, and how it relates to our to those groups. So we're delighted that he could join us, accept our invitation to this symposium. So please welcome Dr. Marcus Bamman. Is it?
I'll turn the lights back on so you can see his face later.
It's here we go. Well, thank you for the invitation, and thanks for the nice introduction. Great pleasure to be here, and I think it's really exciting for me, as somebody who's pretty passionate about this field, to see this type of emphasis and initiative at a place like Johns Hopkins. So it's very exciting. Okay, why am I showing it twice? That's interesting. Let's see if we can just do that. So slight change in my title. The message is very much the same, but I sort of inserted a term here, regenerative, and I'll explain to you why I've done that on the next few slides. This is really sort of a concept that is more than just a concept. I think it's the reality, and I think it's a way for us to embrace the sort of pluripotency of exercise, and I'll dive into that. So we have, as you heard from Dr. Schaefer, a UAB Center for Exercise Medicine, and as part of that center, we have an NIH-funded center that is really focused on helping everybody develop the next and most innovative clinical trial in this field, and that's the National Rehabilitation Research Resource to enhance clinical trials. And many of those trials are exercise-based. So this is my disclosure slide, and it is complete. There's no money in exercise. That's not entirely true, but clearly we tend to be the anti-drug, so that type of money doesn't tend to funnel our direction. So this is our center at UAB, just to give you an idea of what we do and why. Those 4 words, moving research into medicine, is really what we do. And this is a vision statement that we take to heart, and I'm hoping we're doing it in a very effective way. But you see 2 people exercising in the 2 sort of primary modes there with those icons, and then you see a microscope, and then you see a stethoscope. So the progression from left to right has meaning, and that's really what we try to do as a center. So we do have federal funding for research in this area from various entities here, and I think the future is very, very bright for exercise biology and exercise medicine. I think the emphasis on it is growing at all levels. I think the appreciation for not only the health benefits and understanding those health benefits in a deeper way, I think there is a greater appreciation at this point in disease-specific and sort of prescription-based, very dosing-type research that we'll talk about for disease-specific areas. I think that's also growing. And I also think just the concept of this being an innovative approach and an approach approach that feeds very well into precision medicine is growing too. So we have a big group of folks at UAB, some outside of UAB who we collaborate with down the lower right. We do have some multi-center efforts. I'll touch on some of those. But I always like to show this slide up front because sometimes I run out of time at the end. So I'm going to talk to you about these topics hopefully in the next 35, 40 minutes. I will probably zip past a few things along the way. A lot of times I sort of go to these opportunities to speak. I like to bring more than I might share depending on what people are interested in hearing. But this is what I hope to get through today. Tell you a little bit about regenerative medicine, why we think what we do is regenerative, and in the context of that, sort of give you two stories, one in aging and one in Parkinson's disease, okay? So what is regenerative medicine? This is a buzz term. I'm sure Hopkins has a unit, a center, an institute, a something for regenerative medicine. We all do at this point. This is something that every major medical center wants to do well and wants to be a leader in. But I would argue, and hopefully by the end of this session, you'll come to appreciate that exercise itself is a very powerful regenerative tool. So beyond weight management, beyond sort of calories in, calories out, exercise can do things at a molecular and cellular level that are very, very powerful. We're going to dive into some of that. So to provide, repair, replace, restore structures and functions that are either absent or lost due to disease, congenital defect, aging, and so forth. I'm going to tell you that I think exercise is an endogenous regenerative tool that we currently have. This is just an example. This is from a few years ago in an article sort of arguing against the idea that exercise would ever be druggable, because it won't. One or two pathways at a time maybe, but not the pluripotency that we know exists. So as of 2013, we had already revealed a number of signaling cascades that were either turned on or adjusted in one way or another, up or down, by contracting muscle and by exercise in both endurance and resistance modes. And the consequence of that is activation of a lot of stem cell pools that we have, both systemic as well as tissue resident cells that we know now exist in skeletal muscle, in the heart itself, in the brain, and so forth. And I would say that what we're learning is that a lot of these stem cell pools that can provide, repair, replace, regenerate tissues that are damaged, sort of lay in wait and are somewhat dormant until there's a stimulus that says, hey, activate yourself and do something. And we think exercise is one of those tools. So what we do know about exercise to date, really, in a variety of model systems, from the lower animal model systems to some human work, that we do activate stem cells quite readily with exercise training, and we do it acutely. We can certainly modify our gene expression profile quite robustly, and these responses are rapid and can be long-lasting. We induce a lot of cell signaling. There are a number of encouraging papers demonstrating that exercise induces neuroplasticity and some neurogenesis, myogenic activity, which is sort of what you expect because exercise is activating skeletal muscle. Angiogenesis is an important outcome that I'll touch on, osteogenesis, mitochondrial biogenesis, lipolysis as well. So all these things are important. And the last one, even though this isn't a meeting to specifically talk about cancer, I think it's really important to recognize, just in the last 8 years, what we've noticed in terms of tumor biology in the presence of exercise is really, really powerful and exciting. So a lot that we have learned and a lot we will learn. I'm not going through each of these papers individually, but just to give you a flavor in the last few years, you can read them for yourselves. These are really exciting animal studies. These are studies that are telling us that exercise truly is regenerative and has impact on local tissues for activating and restoring functions through primarily through molecular mechanisms as well as activation of local stem cells. I'll just tell you that if anyone in the room is dealing with cancer patients, two of the papers that really get me excited are the last two. And so here we've got a paper that demonstrates improved chemotherapeutic efficacy because by improving vascularization of the tumor itself, there's evidence now that that will improve delivery of the chemotherapy. And if that's the case, reduction in the chemotherapeutic dose potentially reduce side effects and so forth. So that's very, very exciting. This one, strengthening essentially the blood-brain barrier to reduce metastatic cancers, really, really important and exciting stuff as well. So my concept is here. This is the fundamental tenet of what we do. So we are a demand-based organ system. Every one of our organs responds and adapts to the demand that is placed on it. So if we are chronically in a situation of low demand, the result is over time we'll have a low functional capacity and unfortunately a higher probability for some level of pathology, as shown right here. If, though, we are in a chronic state of high demand, which would be the case if you're afterload, if your arterial pressure is such that 70 times a minute, 24/7, your heart is beating against a high afterload in a hypertensive. Now it's a chronic high demand, which is not a good thing, results in fairly low functional capacity and a high probability for pathology. Exercise is the sweet spot. So what we try to explain to people is we want to understand how the systems adapt to intermittent high demand, which is counterbalanced by rest intervals. And what we've found in every system that's essentially been studied is that you improve the functional capacity of that system and you reduce the pathological risk in that system. All right, so that sort of gives you the backdrop for what we're here to talk about. So I'm going to give you a little bit of data that ranges from the most molecular to sort of the most clinical, and hopefully it'll touch each of you based on your own areas of interest. So let me just tell you a little bit about molecular mapping and why we think it's important. And skeletal muscle really as both a tool and a target, I think, is a message I want to send here today. Exercise activates and does things in every cell system that we have studied, but muscle, being the tissue that goes from a truly resting state to a very highly active state, is one that we think plays a major role in the systemic responses as well as the local. So that's just an MRI series that I like to show just to make sure people are awake. All right, so what does muscle look like? I put that up for the medical students, and you know, I guess if you stare at it long enough, you might actually get sleepy. But so when we take a piece of muscle out of a person or an animal, we stain it, and we want to see the fiber types. We want to understand the histological profile and so forth. I just need you need to, if you're not a muscle person, and I have to confess I'm a muscle head, so take it from that perspective. We want to learn about this tissue and how it's adapting and how it's influencing everything else in the context of exercise. So this is just how we identify fiber types. There are three primary fiber types in the human. Type 1 are the fiber types that we have active right now. Low threshold motor units, don't require much intensity to activate those, very fatigue resistant, highly oxidative, and so forth. If the intensity of the contraction goes up, you recruit the 2As and eventually the 2Xs. Interestingly, though, in an exercise training situation, these fibers over here, these 2Xs, the ones that appear in our lab as black, they're not very useful to us. They're highly fatigable. They don't really do much. We actually would prefer to reduce those. These are very high in cases of sedentary status, unloading, obesity, and so forth. We see a lot more 2X. But with exercise training, they shift to 2A and become much more functional, which is a good outcome. All right, so we do this by muscle biopsy. We do multiple muscle biopsies a week. We usually get about 200 to 300 milligrams of tissue in doing that. Very, very useful. So molecular mapping, how do we do it? This is a new study that we're in the planning stages of, very, very exciting, funded by the NIH Common Fund. One of the leaders of that, Dr. Mary Laughlin, is actually here from NIDDK. This will, in the next few years, give us a lot of insight into the acute responses to exercise training at the molecular level in muscle, blood, and adipose tissue in humans, in not only an untrained sort of sedentary state, but also after a period of exercise intervention, and as a comparator, also in some people who are highly trained. So this is a very, very exciting initiative, and we will all benefit from this molecular map down the road. These are just the locations, the sites where various things are happening. There are 12 clinical sites conducting the study. There are 8 molecular mapping or chemical analysis sites doing the analyses, and then we're all, of course, we have a bioinformatics center and a coordinating center. So another study that we're involved in another sort of molecular mapping effort to understand what exercise is doing at the molecular level and sort of relate that to functional outcomes. Is funded by the Office of Naval Research with Tim Broder at Wright State and then Ron Evans at the SALT. And we're doing something quite similar to the study I just showed you, but in this case, I wanted to share it just because we have some interesting sort of data at this point. It's sort of tantalizing. We're doing 2 different exercise doses. These are both involving a combined training stimulus, so both endurance and resistance, but at different intensities. And we're doing a lot of molecular mapping to understand what is happening in response to those modes of exercise. They're training for 12 weeks and so forth. So I just want to share a couple tiny bits of data here, which is very exciting to us. So we think that one of the ways in which exercise is communicating its potential benefits from working muscle to other tissues and organ systems is through the delivery of cargo. And that cargo might be messenger RNAs, it might be microRNAs, it might be small peptides, it might be things that are made in the muscle that can act other places, and they're delivered in these little packages that we call exosomes. So exosome biology is becoming very, very exciting in the exercise field. And what this is basically showing you is that this is just in the first small number of subjects. This study will have 150 people. But it's interesting to me, we get serial muscle biopsies, so before exercise, 3 hours after, 24 hours after. We're seeing a change in the miRNA or the microRNA signature in skeletal muscle, which is interesting. And we're starting to identify things that are not only upregulated short-term post-exercise, which is the red circle, but also long-term post-exercise, which is the green circle, and then a small cluster of miRNAs that are upregulated over that sustained period. We also then do this in the trained state. You can see just by these numbers, when we become more exercise trained, we're more reticent to a response. It's harder to get a response at the molecular level, which is why you can gain strength, gain cardiorespiratory fitness, gain all the things you want to gain early in the first few weeks of training. And then if you become more advanced, gains become slow. We think that's represented at the molecular level as things sort of, it's harder to turn things on and off over time, the more trained we become. So these numbers are small. But interestingly, 4 weeks of detraining, so now we restrict, we pull back the stimulus of the training, and all of a sudden, this microRNA profile undergoes dramatic change. This is a resting sample 4 weeks into detraining. So one message, exercise training is powerful. Second message, detraining is equally powerful. And if we take the stimulus away, we can't expect all of those benefits to be sustained. When we look at the blood and isolate exosomes from the blood and then characterize them on their microRNA profile, very exciting to us, we're starting to identify things that are being expressed in the muscle, packaged up, delivered into the serum, which means they're going wherever the serum goes, which is everywhere. And over time, I hope we will uncover a nice signature of what exercising or contracting muscle is doing to influence other tissues and systems. So that's just a little taste of that. So when, the title was Discovery to Precision Care, what do we mean by that? Well, we've sort of taken the molecular approach to say, what are the outcomes we want to achieve with an exercise training stimulus? How are those occurring? And then how can we take that information to understand why not everybody responds the same? How can we learn from that information to maybe make it more precise or make the prescription more effective for those people that don't necessarily adapt as well as others? So aging muscle, as some of you have probably come to appreciate in your own work, undergoes atrophy. So this is an MRI cross-section of mid-thigh. The feet are pointing out towards you. And by the way, vastus lateralis is this outer thigh muscle here, which is where we do most of our tissue collection. So this is young, this is older, and you can see atrophy. You can also see an increase in subcutaneous adipose tissue, as well as adipose tissue and fibrotic tissue that's sort of invading the muscle. Consequences are pretty serious. I mean, the last one, of course, is the most serious. There's a strong relationship between loss of muscle mass and all-cause mortality. And sometimes we may not appreciate that as much as we should, but it's there. So we've done a lot of studies to investigate and understand sort of how a muscle ages. And so this is a study I'll just show you a little bit of data from that we actually started to understand sex differences as we age at the cellular level. Whole body muscle atrophy in both men and women very similar. At the cellular level, though, it's quite a bit different story. So there's sort of two major ways in which we lose muscle as we get older. One is that the type 2 fibers, which are both those 2A and 2X, get smaller. So we have type 2 atrophy. Turns out, though, that that's more prevalent in women than men as we age. But both men and women lose muscle mass on a whole muscle level, as you should see here. There's no real difference in how that pattern goes. So how do they accomplish this differently? Well, women are clearly doing it by atrophying their type 2 fibers. Men are not atrophying their type 2 fibers nearly as much, which means they're probably losing more fibers in number. And we do know from some very painstaking cadaveric studies, counting every muscle fiber in a human vastus lateralis, that takes time. So we start at a young age with 600,000 to 800,000 muscle fibers in that muscle. Over the span of aging, we lose fibers in number. And so we're losing muscle mass because fibers are going away. We're losing muscle mass because the type 2s are getting smaller. And one of the interesting phenotypes as we age is that we start to see a change in the histological pattern. And this is because as some of these fibers denervate and are on their way to death, they can actually get renervated by a branching axon, which is interesting. And so we end up with fibers that cluster because now they join a different motor unit and become a different type. And I'll get into that a little bit more in Parkinson's. So we're losing fibers in number, we're losing fibers in size, and we need to counteract both of those, and we've done that in a number of different studies. So let me just tell you, resistance training or weightlifting has been our mode of interest because we really want to mechanically load muscle, stimulate hypertrophy, and so forth. So how does it work? In 16 weeks' time, we nearly restore muscle mass to where we want it to be. And these are young versus old. So now we've got people differing in age by nearly 40 years. And we nearly restore muscle mass at the whole muscle level in 16 weeks' time to where the young people were starting. So 40 years of aging almost erased. But at the fiber level, we do. So type 2 fiber hypertrophy, sort of what I call regrowth, results in full restoration. You can also see histologically that those black 2X fibers are all gone. These fibers over here are bigger, and there are no more of these black ones, meaning we've had this nice shift in fiber type. In terms of performance or function of the muscle, strength and power also are largely restored in the older adults. So those are good things. The result of that, and again, the question earlier about standard deviation is really important because those are statistically meaningful results, and they're based on the mean of the group. But what's interesting to us is that we don't all respond the same, and those error bars actually generate some interesting science. So let me just touch on that. So inter-individual heterogeneity is an important concept I want to tell you about. We have, over time, developed this model, and if you were at UAB or even working through the National Resource Center with us to write your next grant application, this is probably the first thing we'd start. So we like to start here. What are you trying to achieve with this exercise intervention? This list is not obviously all-inclusive, but these are kind of some of the main areas. What do you want to achieve? Who are you trying to recruit or study? And what are the potential mediators of whether or not they achieve what you want them to achieve? So we always start here and map out the plan. I'm showing you this today because there are numerous potential sources of why individuals don't all respond the same. And for us to understand those differences enables us to become more precise in how we treat or how we prescribe exercise to certain people. So this is a very important sort of conceptual model. All right, so none of you are allowed to leave this room and say, I'm a non-responder. I don't need to exercise. There's no such thing. We all respond. We just may respond at differing levels based on specific outcomes, which may result in us needing to manipulate a treatment in some way. So this is an example for trying to take older adults and regrow those atrophied muscle fibers. And it demonstrates for you that some people get this very robust response. This is a combination of a couple studies from a whole bunch of people get the response, this sort of 30% response that we expect. and then a fair number of these people don't get much of a response. So we're interested, sort of biologically and scientifically, what makes this group so different from this group? It's kind of interesting. So I'll just jump through a few factors that we've come to learn and appreciate. One is to induce gene expression of things that are sort of pro-myogenic. We know that that happens in a differential way. So this just shows you sort of low or non-responders. They do induce sort of expression of IGF-1. This is actually our first paper where we kind of use cluster analysis to start to tease this apart. And this is muscle level gene expression. There is a muscle isoform of IGF-1 though, this mechano-growth factor, which was differentially affected by whether or not you're a responder or a moderate responder, really high or extreme responder. And then over here, myogenin, which is a transcription factor that modulates stem cell function, particularly differentiation, was also differentially expressed. So that was interesting. And this kind of gave us our first inkling, well, there's something different about how different people respond. One thing I do want to point out, the fiber type shift, that sort of 2X to 2A shift I described, happens in every cluster, meaning they responded, the stimulus was available to them. They just didn't get the hypertrophy and fibers we wanted. So beyond gene expression, sort of digging in, we know that we've got to make more protein to make fiber bigger, and that starts with the initiation of translation. So we've studied this in great detail. I'm going to zip through some of the slides, but let me just tell you that by trying to understand the pathways that lead to making new protein, okay, we've uncovered a couple of factors that are differentially regulated in these different responder clusters, non-responder, moderate, and extreme. And two of these components in that signaling cascade, P70S6 kinase, which is a key kinase that modulates the ribosome, and then eukaryotic initiation factor 2B epsilon, which is also a key kinase that modulates translation. These are differentially expressed, or differentially influenced, in this case phosphorylation, in this case total protein levels, by whether or not you're a responder. And so we pursued that in an animal model. I like this idea of human observation using animal models in sort of a reverse translation to better understand what we've seen. So this is a model where we actually can overexpress something acutely in a mouse or rat muscle. In this case, it's mice. And we really honed in on EIF2B epsilon, overexpressed it, and lo and behold, it does induce hypertrophy. So that told us it's probably pretty important. And we've continued to kind of pursue that. The other part of this, so protein synthesis is one part. The other part of this that's really important is this ability to actually turn on the stem cell pool muscle. So we do that quite well if we are a responder. We do that sort of modestly well if we are here in this middle group. We don't turn on our stem cells well at all if we're not in that group. So a couple points to make. One of the interesting points is that the people who end up being very extreme or robust responders in activating these cells walk in the door with more of these cells. So this black bar is the pre-training bar. They walk in the door with more of these cells than the other groups. I would like to know if I have more of those, right? But that's an interesting sort of story. They come in primed, ready to go, and they turn these cells on quite readily, and they end up with these large fibers. So that's kind of a summary. The low responders don't really activate the protein synthesis machine, and they don't really turn on the stem cell pool. The modest responders kind of do both, and the high responders do both really, really well. So to take that a little bit further, we did some molecular mapping, and this is transcriptomics via microarrays a few years ago. And what we revealed from this, again, here's our three cohorts. Differential gene expression is shown here. So 5,000 genes differentially expressed between extremes and non-responders. That's a lot. Getting to understand that a bit better, we came to this conclusion. The extremes walk in the door primed for muscle response. The low responders walk in the door primed to not have much of a muscle response. And ribosome biogenesis is highlighted because we kind of dove into that. For the sake of time, I'm going to skip a few slides, but let me just tell you that we have identified ribosome biogenesis as a very, very important part of this process. We did this by taking, going into a cell culture system with human primary cells and making myotubes from those cells. And in doing that, we demonstrated that if we, so this, think of this as exercise. If you treat with a very growth factor rich serum, which is what we did, these are myotubes formed from stem cells we pulled out of muscle biopsies. If you treat those cells with a growth stimulus, they will grow. But if you block ribosome biogenesis while trying to get them to grow, they don't. So that was sort of the final step in telling that story. So we interrogated a lot of things to try to understand differential responsiveness and found out that some were meaningful, some were not. So with, a lot of this is summarized actually, sort of mechanisms of how muscle fibers can get bigger. We published this last year. We kind of have a conceptual model that's very useful, I think. It talks about activating the ribosome, making more ribosomes, activating the stem cells, getting those stem cells to provide nuclei to the fibers as they grow. So where do we go from there? Well, I've got this group of older adults that didn't respond so well. So what do I do with them? I give up on them? No, I'm not going to give up. So we approach this in much as you might any other form of medical treatment, whether it be medication or a device or otherwise. What do? We have to identify the optimal dose. We have to identify what is safe, what's effective, right? Everybody wants to know what is the minimum effective dose because you want to avoid the side effects, right? So we did an optimal sort of a dose response trial with 4 different exercise treatments for older adults. And what we originally thought, based on our microarray data and some of the other data, was that some of these older adults who don't respond quite as readily have this inflammatory status in their muscle that needs to be overcome. They also have these stem cells that seem to be sort of slow to turn on. We need to figure out how to fix that issue. So, for the sake of time, I'm going to go fast. 2 of these prescriptions were 3 days a week. The letters refer to how high the intensity was. So here's a group that trained high intensity 3 days a week, so high, 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 Monday, Wednesday, Friday. Here's a group that trained high, low, high. Here's a group that only trained Monday, Friday, so 2 days a week, high, high. And then here's a group that was high on Mondays, low on Fridays. We hypothesized that the groups that were going to do the best were these 2 because they have twice a week high intensity loading of the muscle. And we thought from a functional standpoint, this HLH3 would stand out. By having that additional day, we thought that would be beneficial to them. So consort diagram, bottom line is it takes a lot of work to do these studies. You get 624 people interested. You study 65 people. That's okay. It's a 10 to 1. That's actually not bad for an exercise trial of this duration. These people were with us in total for about 40 weeks. So, lo and behold, we can titrate exercise. We can dose it because what we came away from is that the HLH prescription gave us the greatest gain in muscle mass. These other 2 that also involved 2 or 3 days of high intensity loading gave us a gain in muscle mass, but not as great as the HLH. So that's actually kind of cool. We're now getting to the point where we can titrate the dose. When we look at the thigh specifically, again, which is what we're interested in from a mobility standpoint, HLH gained the most muscle. And we look at strength gain. They all gain strength early on, and then we randomize into these 4 groups. And the strength gains continue to be the most robust in that HLH. So that told us we can dose exercise. Interestingly, when you look at the individuals, again, the spread for gain in muscle mass at the thigh, there is a lot of variability. But what I want you to see is if we focus in on the light gray, the black, the HLH, there's not a lot of HLH over here on the low side. In fact, everybody's gaining something, right? So that kind of tells you why this group did the best overall. Whereas all 3 of these other prescriptions, you got some people over here going south, or some people just not responding at all. So we think we've identified a dose that might be more precise for that population, okay? I'm not going to be able to give you Parkinson's in 2 minutes.
You got to take a few more.
Is that okay?
Yeah.
All right. So let me just tell you about Parkinson's disease and why we're interested. So Parkinson's disease has, first of all, it's age-related. 95% of cases are idiopathic. The motor symptoms are classic symptoms: bradykinesia, resting tremor, rigidity, postural instability. The disease initiates, as most of you probably know, by death of these dopaminergic neurons in the substantia nigra region of the midbrain. And beyond the motor symptoms, there are a lot of non-motor issues of Parkinson's which we study. But I'll just tell you a little bit about the motor first. One of the things that's really interesting, unfortunate but interesting scientifically about Parkinson's is there's this ubiquitous mitochondrial dysfunction. It's not unique to muscle, it's not unique to the brain. It's everywhere that's been studied. And particularly complexes 1 and 4 are not functioning. So if you're thinking about an exercise treatment now, I want to attack the motor system because I want to improve as many of these symptoms as I can. I also want to attack this system to try to reverse some of that. So we've done that. The substantia nigra and what we know about Parkinson's in red here, as a consequence of what the substantia nigra networks do functionally for us, motor symptoms, cognitive impairment, depression, speech impairment, insomnia, REM sleep disturbance. We're studying all these at UAB in the context of exercise treatment for Parkinson's, but I'll just share with you a bit of it really quick. So we decided, let's do something different. There's been a few studies of Parkinson's people in the literature, most of them are walking studies. I want to go high intensity. Can they do high intensity work? So Parkinson's is staged from 1 to 5 on a Honeyard staging scale. We targeted the people we thought would be the most plastic or most malleable, which is a stage 2-3. Okay, so we recruited 2s and 3s, high intensity training, both resistance training and interval training, in a way that was very robust. They're working extremely hard for about 45, 50 minutes. Lo and behold, they do have age-related muscle atrophy. We do correct that in 4 months. They do have age-related strength loss. We do correct that in 4 months. So all encouraging. They do have a depression in terms of their number of capillaries that are feeding muscle fibers, and in 4 months we fix that. They have this mitochondrial dysfunction in particularly the subsarcolemmal fraction of the mitochondria, complex 4, and in 4 months we fix that. All this is pretty cool. We're thinking, well, this is really kind of regenerative. And then if you look at them clinically in 4 months' time, this is the Parkinson's disease quality of life item scale, 39-item scale. Their ADL subscores improve, emotional well-being improves, cognitive impairment subscore improves, total index improves. And then this one, UPDRS, the Unified Parkinson's Disease Rating Scale. If there are any neurologists here or people who work in clinical neurology, you're probably well aware of this in movement disorders. This is a way of clinically tracking the disease. And probably on a semi-annual or annual basis, individuals with Parkinson-like syndromes or Parkinson's disease as a proper will be tracked for their disease progression using this instrument. And we're very excited that in 4 months' time, we're seeing a robust improvement in their motor score as well as in their total score. This 6-point change in the total score, lower is better, by the way, is actually greater than what would be expected or even desired with dopaminergic therapy, L-DOPA type drug therapy. With L-DOPA, over the course of a year, you're hoping that the UPDRS score doesn't change. It tells you that you're at least doing something that's preventing further progression. And here we're actually seeing reversal in 4 months, which is exciting. All right, so exercise in this context we think may be neurogenic proper. And this is an area we're pursuing now. We've got a 2-site clinical trial in Parkinson's where we're looking at this with a deeper dive. But I'll just show you our initial study that gave us some idea or some excitement about this. So if we do this high-intensity exercise bout, what is it doing to the brain? So you use functional MRI, resting state, to look at brain activity, and particularly what this really measures is blood flow to parts of the brain. And then you can quantify. It's basically an index for us that those parts of the brain are becoming more metabolically active. And the good news for us is that the prefrontal cortex is lighting up, which is a very, very important part of our network that leads to motor control, as well as cognitive function. But the substantia nigra, where the disease originates, is lighting up. So we're somehow improving metabolic activity in the substantia nigra. This is 1 hour post-exercise. So we're very excited about this. We think it might lead us down a path to really understand functional connectivity in the brain, how exercise is influencing that. I'm super excited about that. I'll share with you, I showed you in the aging background that there's this concept of denervation, reinnervation leading to that pathological grouping of fibers. And let me just say in Parkinson's, really briefly, it's much more extreme. And I'll show you just a couple slides and wrap it up. So whereas the older adult sees more fiber type grouping, look what happens in Parkinson's. These people with Parkinson's are the same age as these people. And the fiber type grouping, you can see it histologically a lot more easily than you can in the bar graph. This is very much what I would call a pathological state of a muscle. It's not a muscle that has this nice mosaic pattern like a young muscle. So this is aging, and this is aging with PD. Interestingly, though, and it actually has a functional correlate or functional consequence. And when we do something simple like a sit-to-stand task, we use surface EMG to measure sort of what is the relative motor unit activity or the relative intensity of just a simple sit-to-stand. How much of that muscle has to be turned on? And what we see is in the young people, this is a really easy thing to do. As we get older, it's a much more difficult thing to do. And in Parkinson's, it's nearly 90% effort to do this. It's not that they're weak. It's that their motor units are being activated or overactivated to accomplish the task. So it's got this sort of a recruitment pattern that is not normal. So it turns out there's a pretty well-characterized pathway that modulates the integrity of the neuromuscular junction. It involves this cascade and its axis. And I'm not going to show you a lot of that data, and I'm actually going to get my cartoon out of the way as quick as possible. But we studied this in detail to know if the exercise stimulus is actually influencing the synaptic junction between the muscle fibers, between the axon termini and the muscle fibers, to know if we're actually helping to mitigate some of these changes distally. And sure enough, 4 months of training, we start to see that extreme fiber type grouping go down. So when I first saw some of these data from a graduate student, I didn't believe them. I said, that's not possible. This fiber joined a new motor unit. It can't go back. That's not possible. But he kept bringing me more and more data. And our neurology colleagues, we were all skeptical about it. We just weren't sure what to think of it. But we started digging into this, and it turns out that in the animal literature, there's some evidence of dual innervation. A fiber, an individual muscle fiber, can actually be activated synaptically by both a type 1 and a type 2 motor at the same time. We think that's in a transitional state. So our hypothesis, which we're pursuing, is that some of these fibers are being denervated and renervated at the time we start exercising people, and we're convincing those fibers to pull back and stay home. So we've done a lot of study of this sort of cascade that involves the junction, and interestingly, in the Parkinson's, their acetylcholine receptor protein expression levels as well as at the mRNA levels are improving. And then that sit-to-stand task we've normalized in Parkinson's in 4 months' time. So that's kind of exciting for us. So my favorite cartoon, I have to always show you that. Can't get away without showing you that. I will stop there because I'm a little bit over time. If anybody's interested in learning more about our National Center and how you might use that to your advantage, we do have a pilots program and so forth. So thank you.
Questions?
Yes, sir. Mm-hmm. So I didn't show you the data. It was actually on a summary slide, but interestingly, we don't think myostatin plays much of a role here in the adult muscle at least in its response to this type of stimulus. So we looked at it at both protein level and gene expression level, and resistance exercise, a single bout of resistance exercise, downregulates myostatin gene expression in everybody. Actually quite robustly. I mean, it bottoms out. But it's not prognostic or predictive of anything. So it's downregulated in the low responder equally as much as it's downregulated in the extreme responder. We then said, well, what's going on here? Well, let's look at the protein level. Look at the protein level, same thing. So from my vantage point, and I've studied myostatin for a long time, it's a very, very powerful anti-growth factor in the developing muscle. I'm not quite sure any of us understand what it's doing in the adult muscle. Now, there are exceptions to that in certain pathologies where we know that it's playing a role in the adult muscle, but that's because in many myopathic states, we're starting to recapitulate the developmental biology to try to restore and regenerate muscle as it's being impacted by a primary myopathy. But in the normal aging muscle, we're not seeing it. In fact, the pharmaceutical industry has tried to develop ways of blocking myostatin for the purposes of treating aging atrophy and other things. And those aren't really revealing much of an effect. Other questions? Yes, sir.
The question I have for you is about Parkinson's. What is the dose, high intensity exercise?
Yeah, I glossed over that. So they're coming 3 days a week. The resistance training program is whole body training. Because we learned so much in the older adult that doing it high, low, high works extremely well, we adopted that for Parkinson's. So one of the things that we learned from those dosing studies is what to use for the next study, right? So we also just finished an aging study with the high, low, high program, with and without a drug in a placebo-controlled double-blind fashion. That's with Charlotte Peterson at University of Kentucky. That study is in 100 older adults. But for the Parkinson's, we use the high, low, high program. But it's supplemented by body weight intervals in between the resistance sets. So let's say, chest press, leg press, go on down the line. They're not coming off of a machine and just resting. They're coming off of a machine and they're doing body weight something. Step-ups, lunges, you name it. So that we keep their heart rate up, we keep them active, we challenge their balance, we challenge sort of what I call functional power, moving body weight rapidly in different ways. So they actually maintain a heart rate above 60% heart rate reserve the whole time. So it's kind of got an aerobic angle to it. Many years ago, I don't think it's that popular anymore, but some of you probably seen the infomercials for P90X, right? Well, Parkinson's, is PD, so we call this PD90X. These people are amazing, too. I mean, they come in with tremor, they come in with mobility difficulty, and for the 2 to 3 hours post-exercise, a lot of those symptoms go away. And while they're there, they work really, really hard. Yes, ma'am. The answer is no. The question was, do we see a change in falls or fall risk? The answer is no, and it's because of the level of sort of mobility status we recruit. So Parkinson's with stage 2, 3 Parkinson's, they're not really fallers that much yet. They're starting to become more, more at risk for sure. But we don't experience falls during the exercise. They're closely monitored. We've only had actually a couple people with Parkinson's that we physically had to strap. I mean, there's a couple of them where we literally had a trainer with a torso strap going through the exercise facility from station to station because we were concerned about that. But in the older adults, we've studied 60 to 75-year-olds primarily, and then the most recent study with Kentucky was 65 plus. We just are not recruiting fallers. I think it's probably because our inclusion-exclusion. The geriatricians tell me I'm recruiting sort of the super seniors, but That's not the intent, but the programs are intense and we want to understand the biology, so we do tend to recruit a higher functioning group. Yes, sir. Yeah. Yeah, so PGC1 alpha response, if you've not heard of PGC1 alpha, this is a transcriptional co-regulator that is really important for making new mitochondria as one of its functions. It also plays a role in making new blood vessels, or it's just sort of this capillary angiogenesis. In aerobic exercise, it's acutely upregulated. In our resistance model, there's an isoform, or actually it's a splice variant of PGC1 alpha that one group has demonstrated is upregulated in response to resistance training. It might play a role in muscle mass maintenance or muscle mass regulation. We haven't seen that yet, but in aerobic training or endurance training, it's clearly upregulated. It's a really important regulator. We have an investigator at UAB, Glenn Rowe, who his entire research program is centered on PGC1 alpha and PGC1 beta. So we're doing a lot of these studies in mouse models with differing amounts of exercise to dig into that biology. Yeah.